So as we move on through our section of macromolecules, let's take a moment to remember the three things that we need to know about each class of macromolecules. First of all, you needed to know what the name of the polymer is. And next, you needed to know what the name of the bond is that holds each of the different monomers together. So when we considered carbohydrates, the name of the polymer would be a polysaccharide. And the name of the monomer were monosaccharides. So this one's pretty simple. The bond that holds them together, remember, was the glycosidic linkage. So now it's time to diagram our carbohydrate macromolecules. We'll just draw some generic monomers. Make my generic monomers look slightly like 6-carbon sugars, and we link them together in a strand. Each individual hexagon shape is the monomer, or one monosaccharide. They're linked together here by glycosidic linkages. And this whole item itself is the polysaccharide. Now the question is, how are those glycosidic linkages formed? They're formed by the process of dehydration synthesis. So we pull out an H2O as we stick each one of these glucose molecules onto the next one. Now let's begin on our next macromolecule class. Our next macromolecule class are the nucleic acids. The nucleic acids are DNA and RNA. So the polymer name here is simply nucleic acids. The monomers are nucleotides. The monomers in the case of nucleic acids are a little bit more complex, however. You'll notice that these monomers are composed of 3 components. There's a sugar component here, which is ribose or deoxyribose, depending on whether we have a DNA molecule or an RNA molecule. There's a phosphate group, as you can see here. The phosphate group is linked to the sugar, and on another portion of the sugar we'll see the nitrogenous base. So there are 3 components to the monomer, and each of these monomers are strung together to form a nucleic acid. Again, the sugar in deoxyribonucleic acid, or DNA, is deoxyribose that you see here. And in RNA, it's ribose. That simply means we have this oxygen, an OH group, or deoxy, no OH group, on this carbon. Let's orient a little more closely to this sugar molecule. It's a 5-carbon sugar. The first carbon is here, and we note it as 1'. And each of the corners of this molecule represent another carbon, with the 5th carbon being here. There are 3 important carbons for us to consider when we look at a nucleotide. There's the 3rd carbon, the 1st carbon, and the 5th carbon. The reason this is important is the 1st carbon is where we see the nitrogenous base. The 5th carbon is where we see the phosphate group attached. Now, the 3rd carbon contains this OH group. And this OH group is going to be really important for our understanding of how DNA comes together. So let's take a moment to quickly diagram ourselves a generic nucleotide. It's important. You'll need to diagram many nucleotides shortly. So we'll begin with our 5-carbon sugar. There's an oxygen at the peak of it, and then we have this hexagonal shape and a carbon sticking up off of this point. We also know that there's an OH group off the 3' carbon. This one's important for us to note. In addition to that, we'll find off of the 1' carbon that there's a nitrogenous base, a nitrogen-containing base. Off of this 5th carbon up here, we'll also see a phosphate group. So this is a simple diagram of a generic nucleotide. Each of these nucleotides will be connected to each other by a phosphodiester bond. So here is one nucleotide. The simple version, we've got the sugar, the nitrogen-containing base, and the phosphate group strung to another nucleotide, and so on and so forth. Now, the nitrogenous bases vary from nucleotide to nucleotide. However, the 5-carbon sugar and the phosphate will remain consistent. There are two types of nitrogenous bases. There are the purines and the pyrimidines. The purines are called adenine and guanine. You'll notice them here. And the pyrimidines, you'll see there are 3 types: cytosine, thymine, and uracil. Adenine and guanine, or the purines, are both 2-ring structures. You see there are 2 rings here. Whereas the pyrimidines, thymine, cytosine, and uracil, are just single-ring structures. Now, I remember this because the longer name has the smaller structure, as in pyrimidine is a longer word than purine, and there's just a 1-ring structure. And purines have a shorter name and a larger structure, the 2-ring structure. And then, how do you remember which nitrogenous bases are pyrimidines and which ones are purines? Well, I use the rule of Y. Thymine contains a Y, and cytosine contains a Y, and so does the word pyrimidine. So the pyrimidines are made of thymine, cytosine, and then uracil is an outlier because we only see this in RNA, and if you remember that in RNA, uracil replaces thymine, so we see no T in DNA, only U's. So you've got 2 things to keep in mind here. The short name purine belongs with the larger structures, and the long name pyrimidine belongs with the smaller structures. And then the rule of Y's: thymine, cytosine, and pyrimidine. So each of these nucleotides are bonded to each other through dehydration synthesis and the formation of what are called phosphodiester bonds. So now let's diagram an RNA strand. You'll get good at this. We're going to have a pentose sugar, or 5-carbon sugar, with a nitrogenous base. We won't be specific yet, and a phosphate. In addition, the important other component was this 3' OH group. Now we'll string these guys together by simply drawing another pentose sugar with its nitrogenous base and a phosphate. Let's draw a third one, a third pentose sugar with its phosphate, a nitrogenous base, and a 3' OH group. Oops, we forgot the 3' OH here. Now each of these individual nucleotides are held together with a phosphodiester bond that forms between this OH group and this phosphate group. It occurs by dehydration synthesis, so we steal an H from the phosphate as well, and we produce water in the formation of this bond. Again, here, a phosphodiester bond by dehydration synthesis. And here, so now you've diagrammed a single-stranded RNA molecule that contains 3 different nucleotides. In your diagramming assignment, you'll need to draw the other side of this to create a DNA molecule. So let's take a little closer look at DNA, or deoxyribonucleic acid. It's the molecule that encodes information for the amino acid sequence of proteins. We'll look at this later in the semester when we explore transcription and translation. But basically, each of these sequences of bases on one strand of the DNA will code for specific amino acids, and then those amino acids will be strung together to form proteins. We'll be examining the proteins here shortly. So you're probably all familiar that DNA comes in the form of a double helix. Its double helix form is composed of 2 polynucleotide strands. You'll need to diagram a section of DNA for your assignment this week. So you've done the RNA already, and now we're going to stick another upside down piece because they're oriented in opposite directions to each other. So the RNA molecule was one polynucleotide strand, many nucleotides in a row. In DNA, we have two of these polynucleotide strands, oriented in antiparallel direction. We'll look at that here shortly. The two chains are held together by hydrogen bonds down the center, as you can see here, between the nitrogenous bases. In this pairing fashion, adenine pairs with thymine, and cytosine pairs with guanine. So I remember this because GC is my initials, and T and A, well, you can do what you like with that one. So again, to review, each nucleotide is held together by a phosphodiester bond, and each strand is held together by a hydrogen bond between each of the nucleotide pairs. Now go back to last chapter and remember, which are stronger? Covalent bonds that we see in the phosphodiester bonds or hydrogen bonds? If you recall correctly, the phosphodiester bonds are much stronger. So, when you get stress on this molecule, where is it most likely to come apart? Well, it's most likely to unzip right down the middle, and that will have some great implications later on in the semester. Now let's talk about the antiparallel orientation of these strands. As you can see in this diagram, there's a 5' end to this molecule of DNA, and there's a 3' end here. Now, if we look at this one nucleotide at a time, you'll see that we have one nucleotide here, a phosphate, a sugar, and a nitrogen-containing base. The phosphate was located on the 5' carbon, and the OH group was located here on the 3' carbon. So at the other end of this strand, if we follow this all the way around, we'll see that this end is the 3' end, with the 5' end being this direction. Now the other strand is antiparallel. That is, the nucleotides are oriented in the opposite direction. Here's one nucleotide. We have the 3' OH hanging off here, and the phosphate on the other end. So this strand, the 3' end is this end, and the 5' end is over here. That's what we mean by antiparallel. Let's compare this with RNA. RNA is very similar to DNA, except it contains ribose instead of deoxyribose, so it has an extra oxygen. And also we replace thymine with uracil, and it's a single polynucleotide strand. So in this upper figure, you can see a DNA molecule with hydrogen bonds down the center, 3 of them between guanine and cytosine, and only 2 between adenine and thymine. So which one of those is stronger, a triple hydrogen bond or a double hydrogen bond? You're right, a triple. Here we have an RNA molecule, just a single strand. So the nitrogenous bases just hang out there on their own. They're not paired with anything. Which end's the 3' end? Which end's the 5' end? That's a good question for us to look at right now. I'm just going to draw a box around one nucleotide. We've got the ribose, the phosphate, and the uracil. Well, the phosphate is on this end, and the phosphate is attached to the 5'. And then there's an OH group that would be hanging off here. We don't draw it, but you just need to know there is one. So in this case, we have a 3' end, and over here, we'll call this one the 5' end. This will be really useful in our understanding later on in the semester when we look at how DNA is replicated. So RNA uses information in the DNA to specify the sequence of amino acids in proteins. So basically what that means is DNA is the blueprint And RNA is a copy of the blueprint that we take out of the office onto the building site in order to make the proteins themselves. So DNA stays in the nucleus and it's copied into a strand of RNA, and then that RNA will eventually be translated into a protein. We'll look at that in much more detail later in the semester. Now, there are some other important nucleotides. We won't go into too much detail, but you know you've already seen the 5-carbon sugar, the nitrogenous base, and the phosphate group. But what if, instead of having just one phosphate group, you had 3 phosphate groups? Then we would have a triphosphate group. If the nitrogenous base is adenine, we call this adenine triphosphate, which is the primary energy currency of the cell, because these bonds between the phosphate groups are incredibly rich in energy. If this was a guanine, it would be called guanine triphosphate. Those are the 2 most common nucleotides that can become triphosphates. ATP by far, though, is the most prevalent. Another couple of nucleotides we might see are NAD and FAD. We won't look at them in too much detail here, but these are also electron-carrying molecules. We'll see that they have a critical role in the formation of ATP when we look at oxidative metabolism. Okay, so it's time for a card quiz. The addition of water to break a chemical bond is termed red, dehydration synthesis, blue, hydrolysis, yellow, condensation, or green, dissociation. Adding water. So when we add water, do we break it or form it? The monomers of DNA are covalently bonded together. These bonds are called phosphodiester bonds. Green, this is true, or blue, this is false. Which of the following is not part of a nucleotide? The 5-carbon sugar, a phosphate group, a fatty acid chain, or a nitrogenous base? Dehydration synthesis. Green is used to link monomers. Blue removes water to form bonds. Red is involved in protein synthesis. Or yellow, all of the above. If a strand of DNA has a sequence A, T, C, C, T, A, G, what would be the complementary sequence? Pick the right color from below and write it on your paper. Here are the card quiz answers. If you have blue, green, red, yellow, red, you are ready to move on. If you're missing any of these, please go back and review, and feel free to post to the discussion area on Blackboard if you're having a hard time understanding the material. Perhaps I can answer your question there.